Hello everyone. Today we move to another topic of oral sensation. This topic about taste or gustatory system. The objectives include describe the anatomy and nerve pathways of the gustatory system, present the taste qualities, analyze the interaction between saliva and taste, somatosensory and taste. So that the content of our lesson today include anatomy of the gustatory system, basic taste qualities, interaction between saliva and taste, interaction between somatosensory and taste. First, we will discuss about the anatomy of gustatory system. The gustatory system include the peripheral organs. This is also called the taste papillae, the taste neural pathways, and the gustatory cortex. Now, firstly, we will discuss about the peripheral organs. They are taste papillae. The taste papillae are peripheral organs of gustatory system. They include the taste papillae on the tongue and the taste papillae on other locations. The taste papillae on the tongue are protrusions on the surface of the tongue, and they are named according to their morphologies. They include the circumvallate papillae, the foliate papillae, and fungiform papillae. The taste papillae are on other locations, for example, on the soft palate, epiglottis, larynx, oropharynx, and among papillae, there are filiform papillae. They are located on the tongue, but not taste the bud. Circumvallate papilla is located on dorsal surface of tongue between its anterior two thirds and posterior third along the V-line. Foliate papilla is located on the side of tongue, and the fungiform papilla is the largest number, covering the anterior dorsal surface of tongue. They also have extralingual papillae, which they located in the soft palate, epiglottis, larynx, and oropharynx. Another type of papilla is filiform papilla. Filiform papilla is not taste burst, but losing of filiform papilla can cause dry and smooth surface of tongue in some conditions such as dry mouth or in case of anemia. And the functions of filiform papilla is fixing food on the tongue to make the food bolus, and the food bolus can move inside the oral cavity. Inside its taste papilla, there are many taste buds. They are the functional units of peripheral taste organ, and in fungiform papilla contain about 1 to 10 buds. Circumvallate papilla contains about 100 to 200 buds, and the foliate papilla has several hundred to several thousand buds. About the structures of taste buds, they have onion shape, containing 3 types of cells. They are spotting cells at the periphery of taste buds, taste receptor cells continuously mature, located in the inner part of taste bud, and the basal cell, located in the epithelial layer at the base of taste buds. At the germ cell, they can divide, differentiate, and replace dead cells. At the tip of taste buds, the proteins of taste receptor cells are exposed to the oral cavity through an opening of 3 to 5 microns, called the taste bore. The reaction between taste stimulus and taste receptors takes place inside the taste ball. The afferent nerve terminals at the membrane of taste receptor cells at the base of taste bud transmit taste information to central nervous system. About the characteristics of the taste burst. Within hours of exposure to chemical stimuli, the taste receptor cells fall into the oral cavity and are washed away by saliva. The taste receptor cells have a rapid turnover rate, about 10 days, and the taste receptor cells continuously grow and mature, then migrate from the basal region toward the taste ball. Next, we move to the taste neural pathways. There are two processes of taste neural pathways, include the peripheral process and central process. In the peripheral process, the taste stimuli first react to taste receptor proteins at the taste ball. Then the cell depolarization and release the neurotransmitter from the receptor cell to create the potentials so that the taste stimuli are converted into electrical signals. And the electrical signal is transmitted to primary afferent nerves. The information from the taste receptor cells is transmitted by the first afferent neurons, which are the three cranial nerves. The facial nerves transmit information from the anterior two-thirds of tongue, soft palate. The glossopharyngeal nerves transmit information from the posterior one-third of tongue. And the last one is vagus nerve, transmit information from the pharynx, epiglottis, and larynx. The first afferent fibers enter the brainstem and synapse at the second afferent neurons in the taste divisions of a solitary nucleus of medulla. Then the information transmitted through the second afferent neurons ascend to the thalamus. Synapsing with the third afferent neuron in the ventral posterior medial nucleus, and from the thalamus, the third afferent neurons ascends to synapse with the neurons in the region of gustatory cortex in parietal lobe, and the information conscious perception of taste qualities. In amygdala and hypothalamus is also called the limbic region for emotions related to the taste qualities. And in hippocampus, the information saved to memory. About the basic taste qualities. There are 5 basic taste qualities in human. They are sour taste, salty taste, sweet taste, bitter taste, and umami. About the functions of taste. The taste buds assess the benefits and dangers of water-soluble chemicals put in the mouth as the nutrition substances through the enjoyment of eating, the harmful potential substances through fear of spoiled foods or harmful substances. The sour taste, which is excited by proton hydron, The intensity of sour taste is proportional to concentration of acids in contact with taste buds. The sour and bitter taste are protective or warning signals, as protons can have local and systemic effects. Saliva secretion and taste are closely linked, with sour being the strongest stimulus. It increased saliva secretion. When you eat something with sour taste, it can lead to increase the saliva flow, so increase the secret bionates, and which are buffer the protons to protect oral tissues from the damage. And of course, when you have an experience with the sour taste, you will have a grimace reaction with a strong contraction of facial muscles. It also has to push the saliva to the surface of the tongue in order to neutralize protons. About the salty taste, if we consume highlight sodium salts like sodium chloride or sodium bromide, we can feel the poor salty taste. But if we consume non-sodium halide salts like potassium chloride or ammonium chloride, we can feel salty and bitter taste. About the sweet taste is stimulated by many compounds, including some amino acids or artificial sweeteners like saccharin or aspartame. Bitter taste. One of the main roles of bitter taste is to detect potentially harmful or toxic compounds. The stimulant compounds can range from the ion, such as potassium, to complex artificial, such as the denatonium benzoate, or nature like caffeine or quinine compounds. The bitter taste with low bitter perception thresholds. This is the lowest threshold of all taste qualities, where the aversions of bitter taste can have a vital benefit. Umami taste. A taste quality specific for monosodium L-glutamate, which is called umami. This means delicious in Japanese. The monosodium L-glutamate acts synergistic effect with 5-ribonucleotides. Japanese foods often combining pork, chicken, black mushroom, which containing nucleotides with tomatoes, carrots, mushroom that are rich in monosodium L-glutamate, which is synergistic effect of ligation to increase the appetite. All three nerves, 7, 9, and 10, transmit these taste stimuli. Most taste buds respond to 2, 3, or 4 basic tastes, but the majority respond to 1 or 2 tastes. The sweet and salty tastes are found mainly at the tip of the tongue, while the bitter on the back of the tongue and palate, the sour on both sides of the tongue. About the interaction between saliva and taste. The taste functions at peripheral level requires 3 elements: a molecule that elicits taste, a taste receptor cell, and a water environment. There are mutual interaction between taste and saliva secretion. Saliva affects taste. In the opposite way, many taste stimuli affect saliva composition. Saliva play an important role in taste functions. In our cavity, saliva can dissolve taste stimuli and transport them to the taste buds. It also provides an ionic environment to convert taste stimuli into electrical activity, since then conduction of taste signals by the first afferent nerves. The decreasing or increasing saliva flow affects the response of taste buds to taste stimuli, such as decrease or increase threshold for sour taste perception. So that people with local and systemic disorders will be affected to salivary flow and had an effect on the taste. For example, the radiation therapy to the head and neck in cancer treatment can damage the taste cells and saliva glands, and they can affect taste indirectly or directly. In the opposite way, the taste stimulation plays an important role in the excretion of saliva. The taste stimulation affects saliva quality and quantity. For example, the sour taste causes increased saliva flow rate, and it also increases the concentration of bicarbonate, which is the main buffer of saliva. There is an interaction between somatosensory and taste. Taste and smell interact with common sensory systems, include the thermal sensation, mechanical sensation, and pain sensation, to provide an overall assessment of the chemosensory and somatosensory properties of food, which is taken into the mouth, to increase quality of life, and human protect against potentially harmful compounds. So we have just learned about the taste or gustatory system. Thank you for your following and see you next time in the next oral physiology lesson.